Hello Anatomy, welcome to our video lecture for Chapter 7, the nervous system. So Chapter 7 is a pretty lengthy chapter. We'll probably have to divide this into 2, maybe 3 videos. The nervous system is obviously a very complicated system, so let's just jump right in. Okay, so when you think of the nervous system, the word that should come to mind is probably the word communication. Through your nervous system, the outside world communicates with you, and that you can tell what's going on around you, and then you communicate with the outside world in terms of your nerves, and influencing your muscles and telling your body how to respond to stimuli. So the first function of the nervous system, sensory input, is basically the idea that your body can monitor your surroundings. It can tell you temperature, it can tell you what's around you, you can hear, you can see, you can smell, you can taste, you can touch. All the changes that come toward you that your body detects are called stimuli. Integration would be what goes on in your brain or your spinal column, where your body takes all the sensory input that's thrust at you all the time, all day long, and interprets it. An example: sound are just waves going through the air. They're really just vibrations. The sound is detected by your ear, but then your brain is what turns those vibrations into a voice, into music, into a bee buzzing around you. So your body detects the vibrations, then your brain integrates them and basically decides, what is that? What do I translate that stimulus into? Thirdly, motor output would be how your body responds to a stimulus. So usually, your nerves control your body, and that they control your muscles, like to move, or they control glands to give off different chemicals, things like hormones. So sensory input is what comes at you. Integrations when your brain or your spinal column interprets what those signals are, and motor output would be the effect of what was coming at you, right? So, your nervous system is one big happy system, right? We subdivide it into different categories to help us learn it and help us explain it. So the two main sort of classification systems of the nervous system are the central nervous system and the peripheral nervous system. That's how we subdivide it, but it's really just one big happy nervous system, right? The central nervous system, or CNS, is your brain and your spinal column. All the signals that your body receives either are integrated by your brain, or in some cases, your spinal column. So this would be the command center. This would be the system that interprets incoming signals and then turns them into something, and then sends signals out to your glands or to your muscles. Your peripheral nervous system, or the PNS, would be the nerves that extend from your brain and your spinal column through your entire body. You have spinal nerves. We're going to go through them in a later video. They carry impulses to and from your spine. And then you have cranial nerves that carry impulses to and from your brain. The functions, again, these are your communication lines. These are the nerves that detect things and the nerves that send signals from your brain or your spinal column. So this diagram, we're going to go through this in some detail in a minute, but this kind of summarizes everything. Here at the top is your central nervous system, your brain and your spinal cord. The peripheral nervous system would be all the nerves that go through your body. So starting here on the left, your sense organs, your eyes, your ears, your taste receptors, your touch receptors, your nose, they detect signals. Those signals are called afferent with an A. Affect versus effect. You learned this in like 8th grade. Affect with an A is the verb, things that affect you, like you see something or you smell something. And then effect with an E, effect is a noun, like the effects of something. So, if I see someone coming at me, I might step out of their way. The seeing is an afferent pathway. The stepping out of the way is an efferent pathway. These would be controlled by motor neurons that obviously, control your muscles. So you detect signals through your sense organs. Sensory neurons carry them through the peripheral nervous system to your brain or spinal column. Your brain or spinal column decides what to do and sends the signals to motor neurons, which carry out the command. Two kinds of motor neurons, somatic and autonomic. But all the things that we're saying are going to be on slides coming up. Your somatic nerves are the ones that are voluntary. The root soma means body. So these would be mainly the ones controlling your muscles. Autonomic would be the ones that you can't consciously control, the ones that happen without you having to think about it. So things like your heart, things like your glands, smooth muscles, that's part of the autonomic nervous system. And you can further subdivide, and we'll do this more later, the autonomic nervous system into two divisions called the parasympathetic and the sympathetic, and we'll get to those later. So like I say, all the things that I just said are on these slides coming up. So two kinds of structural divisions in the peripheral nervous system. The sensory division, that's afferent with an A, that's what detects stuff. So your nose, your eyes, your ears, things like that. The motor or the efferent division are the nerve fibers that go from your brain or spinal column away to your glands or your muscles, and they carry out the signal. So let's do an example. Let's say you poured yourself a hot cup of coffee and you went to pick it up and it was too hot to touch, so you like flinched away. So you touched the cup of coffee, it was too hot. Temperature receptors in your skin were like, whoa, that's too hot. A afferent nerve with an A detects the heat and sends a signal to your brain, the integration center, which told your hand to put it down or to flinch or to back away from the cup. That signal got sent through an efferent pathway from your brain to your muscles and your hands and your fingers, all right? So afferent are your sensory neurons, efferent are your motor neurons. Obviously, this is gonna be very important in chapter 7. And this, I've already said all this. The efferent division would be the signals that carry out the brain's instructions. The two subdivisions were the somatic, which were the voluntary ones, and the autonomic, the ones that were involuntary. And again, we further divide the autonomic into sympathetic and parasympathetic. Okay, so let's talk about nervous tissue. So there's different kinds of cells in your nervous system. The term neuroglia, so neuroglia, the word means nerve glue. These are not cells that actually carry signals. These are cells that support the nervous system. They might insulate them, they might nourish them, they might be for protection. Again, you know, your brain needs them, but they're not the ones actually carrying out the signals. They're like the support staff. So let's just go through some examples relatively quickly. Astrocytes, so you've seen this picture, this yellow thing is a neuron. Astrocytes, and actually the light red things are blood capillaries. These are star-shaped cells. Astro means star, cyto refers to cell, so these are, the word means literally star cell. These are cells that have projections that kind of grab to neurons, and they can grab a neuron and they can grab a blood capillary. So they support neurons, they brace them, but they also are sort of like the middleman between your blood and your nerves. Nerves are very, very sensitive. Nerves need to be protected very, very carefully. So you don't want things going directly from your blood to your nerves, because, you know, there's a toxin, if there's something that you don't want, that could be bad. So there's a barrier between your blood capillaries and your nerves that's formed by these astrocytes. So say that you have a nerve cell that needs oxygen. Well, that's fine. You get oxygen through your blood. But the oxygen goes from your blood capillary to the astrocyte, then to the nerve. And it's sort of like, again, like a middle layer of protection to make sure that the things that are going in the nerves are, and things that should not be going into your nerves are not. All right. You know, if you have a lot of glucose in your blood, you don't necessarily want to just swamp your nerves with the glucose. You want to give them what they need when they need it. Microglia are these sort of spider-like cells. They're phagocytes. Those are cells that kind of that engulf things. They help protect the neurons. They gather up debris that shouldn't be there. They're sort of like little soldiers that help protect your nerves. Ependymal cells. So you might have heard of cerebrospinal fluid before. There is a fluid called cerebrospinal fluid that sort of bathes your brain in your spinal column. Its function we'll see in a later video. It's very important. It needs to be kept moving. You don't want it to be still. If you ever have a spinal tap, which is rather painful, what they're doing is they're taking some cerebrospinal fluid from the base of your back. If that gets infected, like if that gets meningitis or something, that's very, very bad. Anyway, these cells are ciliated. They have little cilia that beat back and forth, which help keep that fluid circulating. Oligodendrocytes. So nerve cells are very oftentimes wrapped in these, they look kind of like white sheaths. It's this fatty material called a myelin sheath. It's sort of like if you've eaten a jelly roll, you know, a jelly roll has the layers of cake or whatever it is, and there's jelly inside it, and you sort of wrap the cake around the jelly. They wrap around neurons to produce things called myelin sheaths. You find oligodendrocytes. Again, think of it like a flat cell that you wrapped around into a roll. These you find in the central nervous system, so in your brain or your spinal column. Oligodendrocytes. Satellite cells and Schwann cells, these are other kinds of support cells. Satellite cells, if you look in the picture, you can see they surround, this is called the cell body. We haven't done the structure of a neuron yet, we'll do it in just a minute. These are cells that protect their cell bodies. Schwann cells are kind of like oligodendrocytes. They form these fatty myelin sheaths that sort of insulate the neuron, but you find Schwann cells in the peripheral nervous system, whereas on the previous slide, oligodendrocytes were in the central nervous system. Okay, now getting to the actual actors. So neuron is the name for a nerve cell. These are cells that are designed to transmit signals, right? The major regions of nerves, the cell body, and again, we're gonna look at a diagram in a minute, is the main body of the cell. It's where the nucleus, all the organelles are. It's where the cells, it's sort of metabolic center. The processes are fibers that extend out from the cell body. Again, this picture, I know I just skipped a slide. This picture's we're gonna go through in a minute. Other parts of neurons, Nissl bodies is a specialized kind of endoplasmic reticulum that's found in nerve cells. Neurofibrils, nerve cells need to have their shape maintained. So it's sort of like a cytoskeleton that helps maintain the cell's shape, and a nucleus with the DNA and a large nucleolus to make ribosomes. So let's spend a little bit of time with this diagram. This is the one you can imagine you might have to label on a test. So here I have a nerve cell. This is the cell body, all right? It has all the cell organelles. By the way, let me just say now, when nerve cells communicate, the direction of signaling, in this case, is going down the neuron like this. See where the arrows are? So the cell body receives the signal and it sends it down what's called the axon to the axon terminal, which would then send it to the next neuron or to a muscle or to a gland. Anyway, so I have the cell body. The projections coming out the cell body are called dendrites. The cell body kind of comes into this cone-shaped part called the axon hillock, and that then goes into the axon, which is the long wire-like structure of a nerve cell. You see this nerve cell is myelinated. It has the sort of white fatty sheaths. One of these is made from a Schwann cell, so this would be in the peripheral nervous system. The Schwann cells don't always touch one another. You can have little nodes. These are called nodes of Ranvier between myelin sheaths. And at the end, you get to, oh, here, the neurofibrils that help maintain the shape of the cell body. At the end, you have what's called the axon terminal, which is where the signal ends in one neuron and then will get sent to the next one. This diagram, obviously, is a very important diagram. Here, I just see an electron microgram of the cell body of a neuron, and these are dendrites coming off. Okay, so I've already said what's on this slide. So the dendrites receive the signal from what's called a presynaptic neuron, the neuron before it, send it to the cell body. The axon conducts the signals away from the cell body, from the axon hillock down the axon, and then it will go to the next neuron or a muscle or a gland. Again, same diagram. So the direction of signaling is the dendrites receive the signal. Here's the cell body. Then the signal goes down the axon, and then it goes to the axon terminal, and then these would be making contact, well, through a synapse, which we'll get to in a minute, with either another neuron or a muscle or a gland. So the axons, again, I just said they end in what's called axon terminals. So we'll do this in a minute. Axon terminals don't actually touch the next neuron. There's a space there called the synaptic cleft, which is a junction between nerves or a synapse. The axon terminals contain little chemicals. They contain vesicles that contain chemicals called neurotransmitters, things like dopamine or acetylcholine. Neurotransmitters are chemicals that will send the signal from the presynaptic neuron to the postsynaptic neuron. So the myelin sheath, we discussed this earlier. These are like the jelly roll like cells that you kind of wrap around the axon to myelinate them. The Schwann cells made them in the peripheral nervous system. Oligodendrocytes made them in the central nervous system. Little gaps between them are called nodes of Ranvier, and why they're important, we'll see in a minute. So the terms like gray matter, nuclei, white matter, ganglia. So, you know, some axons of cells can go from the bottom of your spine down to your foot. So you can have nerve cells that are 3 feet long. The cell bodies, though, are usually located in the central nervous system, so your brain or your spinal column. Gray matter is what you call cell bodies that have axons that are not myelinated, so they don't appear white, they appear gray. Nuclei are what you just call a cluster of cell bodies. So picture like a cluster of grapes, a cluster of cell bodies that are in the central nervous system, and then the axons that come out. Ganglia are just what you call collections of cell bodies that are in the peripheral nervous system, not the central nervous system. All right. So a cluster of cell bodies in the CNS are nuclei, and the PNS are ganglia. A lot of vocabulary in this chapter, right? So tracts versus nerves. So again, picture the cluster of grapes, the cell bodies. They all have axons that come out. A cluster of nerve fibers, if it's in the central nervous system, they're called tracts. If it's in the peripheral nervous system, they're called nerves. White matter, again, is what you call axons that are myelinated. They actually look white, and gray matter would be ones that don't have the myelin sheaths. They look gray, and then would include those cell bodies too. Okay, so this stuff we talked about at the beginning. So different kinds of neurons, sensory neurons or afferent neurons are ones that detect signals. So your eyes, your ears, your nose, tasting, touching. Cutaneous sense organs would be ones in your skin. Proprioceptors are ones that would be found usually in your muscles, and those are ones that detect like tension or stretch. It's how your body knows. Like if you're pushing against something, you know, you might have a skin receptor that detects the touch, but your muscles can actually detect like the pressure. And again, those kinds of neurons detect things. Motor neurons or efferent neurons are the ones that send signals from the brain or spinal cord to a muscle or a nerve. So afferent detect, efferent respond. This just shows what we've been talking about. So remember when I did the hot cup of coffee example? So here I had dendrites that detect the coffee was too hot, sent the signal through an afferent nerve to your spinal cord or your brain. It decided what to do with it. It said, ooh, that's too hot. What should I do? I'm going to put the cup down. Then it sent a signal to your hand to put the cup down through an efferent neuron that went to a muscle. These next couple of images you don't need to know. These just go through some different types of receptors. That's an awful picture, right? You have receptors that detect pain, things like in your skin. Meissner's corpuscles are ones that just detect touch. These ones like this are ones like here. So here I'm like, is that a sponge? Like when I push on the sponge or whatever, or that rubber ball, whatever it is, I can detect pressure. Those are deeper receptors than say ones that detect touch. Here I have a proprioceptor. Like if I'm holding that barbell, like say nothing is moving, I'm just holding it. There's still tension in that muscle, right? You can detect that in this one too. This is the same idea. Another receptor that can detect tension in a muscle, letting you know that it's contracting even though it's not actually moving anything. Those examples you don't need to know. It's just useful to go through different types of sensory neurons. So interneurons, these are ones that connect afferent neurons to efferent neurons. These would be the ones that integrate the signal and, for the brain to decide what to do. These are ones that are found again in your brain and in the nerves of your spinal cord. So structural classification of neurons, let's do this kind of quickly. So the picture that's shown here is a sort of a standard stereotypical neuron. These are called multipolar because the cell body has multiple extensions. Here I have tons of dendrites, and then the the axon hillock branches into one axon. This is a very common structure. Bipolar, here's one where I have, and these are not common in adult humans. I have the cell body. I have one that comes out here that branches into, or one axon that comes out. This part's not unusual, but I have one that comes out and branches to the dendrites here. So whereas in the previous slide, I had a bunch of extensions coming from the cell body, here I only have two. They do further branch later. That's called a bipolar neuron. Unipolar neurons, and these are also not as common as the multipolar ones. I have the cell body, I only have one process that comes out, and then it branches into one side where the dendrites are, then one side where the axon would be, okay? So functional properties of neurons. So now we're gonna switch gears and we're gonna get into how neurons actually work. So neurons have irritability, which means they can respond to a stimulus, and they have conductivity, which means they can transmit a signal. So what I want to do, and this, you probably did this in 9th grade, which is maybe sounds a little bit familiar. I want to go through how a neuron actually works. A lot of times in science, you use the terminology of a neuron firing. Like when a neuron sends a signal, it fires. It doesn't actually fire. What it does is it sends an electric signal. It's what's really called an electrochemical signal. In this case, we're going to do the electro part. But it sends a signal down the neuron that is essentially electricity, all right? It's an electric signal. Neurons at rest, so let's do an example before we talk about this image. So let's say you had a slinky, you had a coil, like a slinky, and you took the coil and you like compressed it, all right? You compressed the coil or a spring, you compressed it down. It took energy to do that, right? It's sort of like if you have a gun and you cock the gun. Doing that took energy, all right? This is called a resting neuron because the neuron's not firing, it's not doing anything yet, it's not sending a signal. But to get the neuron at rest, you had to use ATP, all right? If you look at this picture, this is the neuron, this is the inside, this is the outside. This neuron is what is called polarized because the inside compared to the outside is negatively charged. Now, if you look at the diagram, look at it closely, I have two ions. I have sodium ions, which are positively charged on the outside. And on the inside, I have potassium ions, which are also positively charged on the inside. So there are no actual negative charges, right? But there are more sodiums than there are potassiums. So relatively speaking, the inside is negatively charged in comparison to the outside. It's polarized, even though nothing is actually negatively charged, right? It's just the outside is more positively charged. Does that make sense? And again, here we're gonna have concentration gradients because the sodium that's outside wants to come in because of diffusion, and the potassium that's inside wants to go out because of diffusion, okay? So polarizing the neuron took ATP, it took energy to get it to the state where the inside's negative in comparison to the outside, which is positive, all right? And notice sodium's on the outside, potassium's on the inside, all right? I'm sure at some point someone's taught you the sodium potassium pump, the salty banana, which we'll talk about in just a minute, but the sodium potassium pump is the one that its function polarized the neuron. So let's say that neuron receives a signal, all right? And that neuron needs to send that signal somewhere. What happens is that stimulus depolarizes the neuron's membrane. And what happens here, if you look at the picture here on the left, there are membrane transport gates, which are usually closed. And there's a gate that allows sodium to come in, and those gates actually open. What that stimulus did, maybe it was light, maybe it was sound, maybe it was taste. But what the stimulus did was it caused a change in the membrane and it opened the sodium gates. Now, if the sodium gates are open, what's sodium going to do? Sodium is going to diffuse inside the cell. That's what's happening right here. And when sodium comes in, it makes the left side of that neuron, in comparison, positively charged because sodium is positively charged. All right. So this begins a depolarization of that neuron. Now, look at this slide. Like, the signal keeps getting bigger. Excuse me. What happens here is that if the signal is large enough, its effect causes other sodium channels to open, and the whole left-hand side of the neuron becomes depolarized. And that's called an action potential, because what happens is if you get to a certain electric charge, what happens is that signal is going to go all the way down the neuron from left to right. All right. And that, the signal going down the neuron is called an action potential. In this picture, you can see it's propagated. In sequence, left to right, the sodium gates open, sodium rushes in, which creates that depolarization, which is essentially just an electrical signal that moves down the neuron. Neurons that have those myelin sheaths, the signal goes faster for reasons we'll see in just a minute. Now, one thing that I haven't told you yet. So let me go back. So sodium is rushing in right here, right? Well, once the signal gets, say, to here, there's another effect. And the other effect is that those sodium gates close, but then potassium gates open. If you remember, potassium was on the inside. If I open a potassium gate, the potassium diffuses out. That's what happens on this slide, which is the slide on repolarization. So as the action potential moves to the right, all right, where it previously was, the sodium gates close, so sodium can't come in anymore. The potassium gates open, and potassium diffuses out, which sort of resets the left-hand side of the neuron to be negatively charged. All right. And again, it goes down the whole length of the neuron. Now, once you're done, let me go back a second. We started out with the resting neuron with sodium outside, and potassium inside, and sodium came in down the neuron sequentially. That, the signal that was sent was like a local depolarization of positive charge going down the neuron. All right. Now, by the end, we open the sodium, we open the potassium. We're back to it being negative on the inside, but my neurons are in the wrong place. Remember, sodium came in, potassium came out. How do I reset the neuron back to the way it was? Well, analogous to cocking the gun or compressing the coil, the sodium potassium pump, the salty banana, if you look at a picture of it, so here it is, when it sort of does this number, right? When it opens and closes, 3 sodiums go out for every 2 potassiums that come in. All right, remember we said at the beginning, there was more sodium outside than potassium inside, which is why the inside was more negatively charged in comparison. The sodium potassium pump in this slide, let me go back a second, is what sort of restores the original configuration of the resting neuron back for it being polarized again. So again, to prepare the neuron for firing, takes ATP, takes the salty banana. Salty banana because the sodium potassium pump, sodium is salty, potassium is in banana, so you call it the salty banana. If you haven't heard that before, now you have. That took energy to sort of get the nerve ready to go. To fire the nerve, to get it to send the signal, took a stimulus like sight or smell or whatever, or touch. That initially opened a sodium gate that started the local depolarization, which went down the neuron in what's called an action potential. Okay. Now, that's half the story. So I told you a minute ago, nerve signals are called electrochemical signals. The electro part we just talked about, right? That's done. Once the signal gets to the end of the neuron, to the synaptic terminal, neurons don't actually touch one another. If neurons actually touched one another, a signal could go from the brain to your foot. There's no way of turning it off, right? They don't actually touch. There's a space between the neuron. This is called a presynaptic neuron. This is the postsynaptic neuron. There's a space that's filled with just interstitial fluid that is called the synaptic cleft. How the signal goes from neuron to neuron, and we briefly discussed this when we did the chapter on muscles, is through what are called neurotransmitters. At the end of the presynaptic neuron's terminal, you have these little vesicles with neurotransmitters, these little green dots. They get released into the synaptic cleft. They bind with receptors on the postsynaptic neuron, and then that binding opens sodium gates of the next neuron, which sends the action potential down that neuron. Now, these neurotransmitters, these little green signals or green chemicals, don't live there forever. Eventually, they get broken down, and when they get broken down, the signal gets turned off, right? this just goes through the steps. So the way that these vesicles fuse, you need calcium ions. Calcium ions rush in. They cause the vesicles to fuse. You need calcium too. When the vesicles get released into the synaptic cleft, they will bind with the receptors on the postsynaptic neuron or the receiving neuron, and then they'll, of course, send the signal down that neuron. Okay, we talked about that. Here you can see it's binding. Here you see these, like say this could be a sodium gate. Here they're closed. The neurotransmitter binds to it, opens it, and then of course that causes sodium to rush in and creates another action potential. Yep. Discussed that already. Fantastic. Okay. So again, this is an example. The neurotransmitter binds, opens a sodium gate, sodium rushes in, you create a new action potential. Now, the neurotransmitter doesn't live, if it's there forever, there's no way of turning off the signal. Eventually, the neurotransmitter gets broken down and gets reabsorbed by the presynaptic neuron, which, if it's not there anymore, the sodium gates close and the signal stops. Okay, so we're going to stop there. That was plenty enough for one video. Hope that was helpful, and I will see you guys next time.